Thank you very much for coming to Cold Spring Harbor Laboratory for our initial meeting of biological data sciences. My name is Michael Schatz. I'm an associate professor here in quantitative biology. My lab is actually just up the hill. The common theme of my research is large-scale data, biological sequence analysis, so things like whole genome assembly, whole genome alignment, short-read mapping, variant calling, transcriptome analysis, that sort of thing. We're very pleased to have you here. I would like to next introduce one of my co-chairs, Ann Carpenter from the Broad, so that she can say a little bit about her research as well.
I just wanted to introduce myself because I'm not giving a talk or a poster, and in particular, so I work at the Broad Institute of Harvard and MIT, and unlike a great number of people at the Broad, I don't work on genome sequences, so I wanted to say hello. I work on high-throughput image analysis, and in particular, over the next few years, we have some very tremendous data sets coming online. I'm actively recruiting postdocs, so I wanted to say hello so you can see my face, meet me at lunches if you're interested in integrating functional genomic information from a lot of different data sources. Definitely seek me out at one of the breaks. Thanks.
Our third organizer was Matt Wood. Just about 24 hours ago, he sent me a panicked email saying that his wife had gone into labor 6 weeks in advance, so unfortunately he will not be able to join us tonight. So I think the program committee is a testament to the sort of diversity of people that we hope that would come here. You know, we have different academic disciplines, industry contacts. I know there's a number of people from government here as well. So thank you very much. I know. And I also realize for many of you this is your first time to Cold Spring Harbor Labs, so let me welcome you here as well. So I thought since this is the first meeting of the biological data sciences, I thought I could give just a little bit of the history about how this meeting came to be, a little bit about how the schedule is organized, and then make a few other introductions before we really kick off the content. So this meeting really started just about a year and a half ago. As many things happen at Cold Spring Harbor, I was in Blackford at lunch, just having casual conversations with some of my coworkers here. And to their credit, so Jason Williams, who works in some of our outreach at the DNA Learning Center just across the harbor, and Charlotte Lambert, who works with David Stewart in the Meetings and Courses program here, they kind of approached me saying, well, you know, at Cold Spring Harbor we have a number of fantastic meetings about biology, about different techniques, different molecular techniques. We have some meetings such as Genome Informatics that touches on some of the more analytical, computational, and quantitative nature of biology, but we don't really have a proper home to proper data science. At the time, this is about a year after some of the headlines had come out in, say, Science, really articulating about this trend across biology, but across all really scientific disciplines, about how there's just more and more data being generated. But I think a key point about this is, yes, there's tons of data being generated, and yes, it's interesting, but these data in themselves do not answer any of the questions that we're really interested in. They don't actually answer in themselves, how my genome compares to yours, how activity inside of cells, such as expression or methylation or chromatin change during development or in different disease states. They don't tell you anything in themselves about what diseases you're at risk for, pathogens you've been exposed to, what medicines that we should give you. They don't say anything. They're just raw bits sitting on a hard drive. So I think what gets lost in some of this discussion is, who will actually answer all those questions and how will they do that? And that's what this meeting is all about, is the who and the how. Now, the who part about this is actually the easier part to answer. That's all of us. So welcome. You're all now designated as data scientists, all designated as biological data scientists. So I really like this figure from the Wikipedia where they try to draw a map about what data science is. In the middle, that's where that is. But if you think about all of the disciplines that we connect to, there's computing, there's statistics, mathematics, and then also, of course, there's the domain expertise of biology itself. All have to be integrated together into sort of one coherent picture if we're going to be able to make any advances in the field. I especially like that they drew a special bubble around the hacker mindset. I can't tell you how many projects where what I really bring to the table is my ability to diagnose weird artifacts in data and say, oh, this is real, or this is just some bizarro thing going on there. So I really like this map of data science. This is really a map of who we all are. We all come from very diverse backgrounds. We have different ways of thinking about problems, but hopefully we can all bring you here together so we can talk to each other, share new ideas, and have new relationships. So that's the who. The how is more subtle and more complicated. So I think I'm going to have to decompose this into a few parts. So I think the first response to the how is to talk a little bit about the nature of the data that we're looking at. So clearly, we're data scientists. We're going to be looking at large volumes of data. A really strong example about this of biological data is going to be the rise of sequencing. So there's a new instrument, a relatively new instrument from Illumina called an X10. And it can sequence a whole human genome in about 30 minutes, once you sort of amortize all the costs there. So every 30 minutes, here's a whole new genome, here's a whole new genome, here's a whole new genome. To date, there's something like 100,000 whole genomes that have been sequenced. It's very hard to get precise numbers on these, so I apologize, but I think that's about the right order of magnitude. There's been some announcements from Illumina, and over the next year, they expect several 100,000 whole genomes to be sequenced. If you take all the instruments in the world and you try to add up how much capacity they have, it's something like 25 petabases of sequence generated every year. This is actually a huge number. It's very hard to get your head around as to what that actually represents. So let me try to lay that out for you. So a petabyte is a funny unit, but everything in sort of computers works by factors of 1,000. So we have byte, kilobyte, megabyte, gigabyte, terabyte, petabyte. Still quite abstract to see that many zeros in a row. Let me try to make this even more concrete. If we were to take that petabyte of data and then try to write it out into DVDs, this big, 1.2 millimeters thick, each stores about 4.7 gigabytes. If you were to write about a petabyte of data out to a stack of DVDs, it would take something like 200,000 DVDs to be able to store all this data. Collectively, that would be enough information to store the raw data for sequencing 10,000 whole human genomes. If we were to take that stack of DVDs and then just stack it up in a big tower, it would be about 1/6 of a mile tall. It would be about as tall as the Empire State Building. It's quite a substantial pile of data. Of course, you would never actually write it out to a stack of DVDs. You're going to store it on, in some data center on some big cluster of hard drives. So, yes, a petabyte is a lot of storage. It's $500,000, but it is quite now necessary, in fact, to have capacities of that type. In fact, just up the hill here at Cold Spring Harbor in our data center, we have about 5 petabytes of storage. Just racks and racks and racks of machines. Now, the amazing and also scary part about biological data sciences is we're just getting started, right? So historically, for the last several years, the rate of growth, the rate of sort of data production has been growing at about 3x per year. So if here we are today, it's something like 25 petabases a year. If we just forecast a few more years into the future, sort of anticipating improvements to throughput, selling more instruments, things like that, in around 2018, we should expect to hit the milestone of about 1 exabyte of sequence generated every year. We're starting to talk about very, very large numbers. Now we can play this game into the future even further. It gets very murky as to exactly when and if these technologies will continue to advance that far. But just for fun, if we extend this out another few more years, something around about 2024 is when we can expect to hit the next major milestone of about a zettabyte of sequence produced every year. So this is a very foreign and abstract and weird number, so let me try to break down what that is to you. So this is a zettabyte, right? So it's 1,000,000 petabytes all stacked up. This would be a very large data center. It would be very, very expensive. Maybe the NSA, I don't know, maybe there are some people in the world that have this. This is much larger than even what Google talks about. So let me try to drill this down into what this means to biology. So again, if we think about this stack of DVDs, now we're talking about something like 200 billion DVDs, enough storage for about 10 billion whole genomes. Now, that's more than the population of the world, but that's okay because there's lots of different species in the world that we'd be interested to sequence. In addition, as I'm sure many of you know, a new trend has been to do single-cell sequencing. So you can imagine, maybe from just 1 billion people, we'll take 10 cells, or from 100 million, we'll take 100 cells. So even though it's a huge number, I think there will be demand for that scale. Just to make this a little more concrete, again, if we're going to have this tower of DVDs, suddenly we're like 150,000 miles tall, about halfway to the moon, in this enormous stack of DVDs. So suddenly, genomics is taking on a whole new scale. Suddenly, we're at the scale of radio astronomy or YouTube. Currently, both of these disciplines are also at around 100 petabytes. It's hard to get precise numbers. Also around 100 petabytes of storage, and also growing exponentially. At this scale, the exact value of your exponent really matters as to when you hit these next milestones. So it's kind of anyone's guess as to which one of these three major disciplines is ever going to reach a zettabyte and who will reach it first. And sometimes it doesn't matter, right? The key point is the data are enormous, and they're just going to keep growing and growing and growing. The actual number is just a line in the sand. In addition to this, and in great contrast to, say, YouTube, where everything gets localized to YouTube, or even radio astronomy, where there's data centers built just to support the telescopes that are next to, sequencing is everywhere, right? So here's a map from this great website that keeps a track, that tries to keep track of where the instruments are all over the world. Notice there's sequencing capacity on every continent except Antarctica, although recently I heard about some people that had embedded sequencers onto a ship. So now I guess you could sail that around and do sequencing literally everywhere. And I think what I think the reason I bring this up is it just changes the landscape, right? There's no way that we're going to get everyone that runs one of these sequencers in the same room to all agree on exactly what they're going to do. There's just so much diversity. In addition, there's political boundaries, there's geographic boundaries, language boundaries. Somehow we all have to work together, despite the fact that we're in constant conflict, constant challenges of just the dispersed nature of the data. In addition to sort of the sequencing capacities, the analysis is basically taking place everywhere, right? So there's people from their laptop up to major supercomputing centers are participating in biological data science to be able to make sense of these data. Of course, though, there are certain major hubs that are being established. So in the Northeast, obviously, the Broad is a major hub of production and analysis. New York Genome Center is a major hub. On the West Coast, we have places like CG Hub that are aggregating together more and more data. So I think today this is very much just a cartoon of what I think is going to happen. But I think we're going to see more of this, where the big centers will continue to get bigger and bigger and bigger as they aggregate together larger and larger data sets. And then, of course, they have to work together. And there's some major challenges in the level of sort of computing and networking and just interoperability and all the APIs to be able to make that happen. I don't know if anyone really has a clear vision how that's going to happen. So in addition to this, I've been talking a lot about sequencing. And yes, a lot of this capacity is used to sequence genomes or exomes of individuals. But of course, biology is much more than genomes. If you peek inside of cells, there's so much activity going on, right? So there's different amount of transcription, there's different amount of protein binding, there's different amounts and changes to histones, to all these other epigenetic markers, to the way the DNA folds up. The amazing thing is that through sequencing now, there's all these different assays that can be used to tease out all these different characteristics to look at all this molecular behavior inside of cells. Of course, though, biology is much more than just sequences and just sort of this resolution. And there's all these other diverse data types associated in biological data sciences, all of the imaging, phenotypic data, in the level of NIH exposure and clinical data as well. I guess my favorite anecdote now is how some of the big agricultural firms now are flying drones over their fields to be able to take digital pictures of the crops growing, to be able to, in a very quantitative way, measure the changes to those crops in response to different weather, different soil conditions, different fertilizers. All these things is all becoming incredibly quantitative in diverse data types. So that's a bit about the how. As we look around the field, kind of the next part about this is the what. And this is where things are getting very exciting, right? So suddenly, thanks to all of these technologies, we have the power to look inside of people. We have the power to look inside of other model species, like Drosophila. We have the power to look inside of different disease states like cancer or autism. We have the power to look inside of crops or a microbiome to be able to piece together, well, what are the underlying mechanisms? What is the actual biology that is driving this? So my congratulations to you. My congratulations to all of us for being able to advance biology through these studies. The part of this that gets left out sometimes is that behind these major headlines are entire armies of scientists, data scientists in particular, that are developing new techniques to be able to support this research. So just about a week ago, Nature had put out this list of the top 100 most highly cited papers of all time. I was very pleased to see that among the very top of this list was the Clustall W paper with more than 100,000 citations. It's just being an incredibly big milestone, an incredibly big achievement in the field as a method, and I'm quite proud of that. Other methods are looking at statistical methods. Very recently, there's been new methods. So here I've just picked the MapReduce paper as an exemplar about some of the new, really creative uses of computational systems to be able to support all the data science going on. Now, some of this does come with a risk. I'm sure many of you are familiar of this paper from Yaniv Ehrlich's lab about a year ago. And they came to this realization that as we're producing all these data, not only are we distributing it for scientific purposes, there is a concern there that those data can be mined to be able to, in particular, to take what was supposed to be anonymous data and then figure out who the individual is. I was very intrigued by this paper, so actually this last summer I had an undergraduate student try to reproduce this work, download the data, run the right programs, connect the right databases, and indeed, you can actually de-identify anonymous data following this way. So I think this is a little bit of a caution of as we're producing these data, as we're producing these methods, we have to have some privacy and security built into it. Furthermore, there was a study from Carnegie Mellon a couple years ago, and they actually realized that things that we thought were secure are just not secure at all. So a good example of this is Social Security numbers. You would think this long digit would be impossible to guess, but indeed there's actually a lot of structure to how those numbers are assigned, and they had done a nice study being able to say with just a little bit of information about where you were born and how old you are, they could predict every single digit of your Social Security number in many cases. A lot of this came from what they call the privacy hidden costs of widespread information dissemination. So this is something that we're going to have to really pay attention to as data scientists, how we're going to be able to safeguard all of these data. So again, with respect to how, there's a lot of requirements. We need integration of multiple data types, massively scalable, geographically distributed, incredible flexibility as to what type of data we're looking at, how we're going to analyze it, tolerant of noise, errors, artifacts, support exploration, ambiguity in the questions that we're asking, and then reliable, reproduce, and secure research. So there is no single product that meets all of these requirements today. That's why we're all here together to be able to talk about, well, what would such a system look like that met all of these requirements? So here's my vision. There's other ways of representing this. So here's my vision about how we're actually going to achieve all these goals and answer these questions. It's going to be through an entire stack of technologies. So at the very base of this are going to be our sensors and our storage systems to be able to collect data. As it moves up, it'll go through various compute systems to be able to do some first-pass analysis of this, very clever algorithms to be able to piece it together, sample from it, index it, or whatnot. Towards the top of this is when we get into the machine learning approaches, where we'll be able to look for patterns at the data. And then finally, at the top of this pyramid, this is when we'll be able to establish, oh, here are the genetics of this disorder, or here's the changes to your microbiome in different environments or response to different treatments. So it's a very exciting discipline to be in, but requires just this enormous technology stack. And I would argue there's no single person on the planet that is really expert at every level of this stack. So from that, and kind of my interpretation of how we're going to make progress in biological data sciences, we organized the schedule to more or less mirror exactly that pyramid of technologies. So we have sessions more or less dedicated to data and data mining, sessions dedicated to the compute infrastructure, to the algorithmics, to the biological software, and then finally to the human biology, as just one example of the top of this pyramid about what can we save from all these data. In a very interesting way, I just took all of the abstracts that you all generated, I ran it through a word cloud generator, and clearly data was the star of this meeting, so I was very happy to see that. In addition to the regular sessions, I'm very pleased about the sessions that have been put together. I really congratulate and thank all of the session chairs. They were the ones that were really responsible for selecting which abstracts would be presented as talks versus would be presented in posters. In addition to this, we have a few very interesting things and I hope quite unique and special things about this meeting. So one special thing was, as kind of in addition to just standard sort of research talks, on Friday afternoon, we're going to have what is the first of what we're going to call a master lecture. So Kirsten Lauder from Microsoft Research was very generous to donate her time to be able to come here and basically give us a 101 lesson on homeomorphic encryption and how we can use it as a tool to preserve privacy in genomic computation. For those of you that don't know, this is one of these very clever but complex number theory ideas where you're going to be able to do certain computations on encrypted data, whereas you never actually have to decrypt it. So in theory, this could be a really powerful way to be able to preserve privacy of data in the sense that you never decrypt it, but still be able to work on it, be able to do research on it. So Kristen's going to give us a lecture about how that works. We have one change to the schedule, unfortunately, which is actually just a testament to how relevant all this work is. So Eric Paraxelis was supposed to be a session chair in the human biology session on Saturday. Unfortunately, though, he's a researcher at Harvard Medical School. Unfortunately, he was just tapped to establish some new emergency Ebola clinics in West Africa. So he called me the other day to say, unfortunately, he wasn't going to be able to attend here because they're very urgently trying to set up this new resource. So I said, of course, Eric, God bless you for doing that work, and we really wish you the best with that. As a result of that, I'm hoping that Mark will be willing to be the session chair for the whole morning. We'll just break 20 minutes early, so it's not a major change to the schedule. And then finally, the last thing I'm very excited to announce is really the star of tonight. Not me, certainly not me. The star of tonight is David Haussler, has agreed to give us the first keynote for the inaugural meeting of biological data sciences here at Cold Spring Harbor. I can think of no better person in the world to give the keynote tonight. So David, as I'm sure all of you know, is a very leader in genomics for the last many decades. So he completed his PhD in computer science from the University of Colorado at Boulder in 1982. Since then, he has spent most of his career at the University of California at Santa Cruz, where he's currently a distinguished professor of biomedical engineering. In addition to that, he is a Howard Hughes Medical Institute investigator. In addition to that, he is the scientific director of UC Santa Cruz Genomics Institute, in addition to several other institutes at UC Santa Cruz. He is a member of the National Academy of Sciences and the American Academy of Arts and Sciences, a fellow of AAAS and AAAI. As I'm sure you all know, his research combines mathematics, computer science, and molecular biology, pioneering the use of HMMs and other machine learning techniques for analyzing biological sequences. He was a major player in the Human Genome Project, and I'm sure we all know him best through his work in the UCSC genome browser, being able to provide those data and be able to do interpretation of those data that has empowered so many of our own labs. Recently, he has focused on understanding and fighting cancer, and then also sharing data through the global alignments of genomics and health. Thank you very much for giving the keynote tonight, and I'm really looking forward to the talk you're about to give. Thank you very much.
Thanks so much, Mike. It's a pleasure to be here, and your comments are overly generous. Your introduction was absolutely perfect for what I want to say. Of course, you asked me to talk about the data science issues, and of course, when we're on the same wavelength there, and so this will be very much echoing the themes that Mike just brilliantly introduced. So I want to start with cancer as a motivator. Many of you know that there are some exciting knowledge and results that are obtained from looking at the molecular nature of cancer. For example, we learned a while ago that cancers carrying the BRAF V600 mutation, such as this metastatic melanoma, are treatable by new drugs that actually will kill cells that have that mutation. And you got some of the most spectacular responses that were ever seen in these patients. Unfortunately, the cancer is more complicated than that. And what's happening inside the different tumors is a battle between one or another clone of the cancer to grow faster than the other one. And when you start to treat, a resistance emerges. Just like you would in a population in an ecosystem. So what happens in this patient after 23 weeks of therapy is that most of the tumors come back. And if you actually look, there's like a map of these tumors. Most of them have billions of cells, and you do the back of the envelope calculation, that's enough so that you would actually expect resistance to emerge spontaneously through mutations or through selection on standing mutation at the time that therapy is started. So unfortunately, this is repeated again and again, and it actually has a predictable time course. This is very reminiscent of what we saw with AIDS. One drug didn't work because resistance have developed too fast. So once you get to a drug cocktail of 3, then you have enough coverage to block. And that was kind of the magic number there. There will be numerical magic numbers for these things for the treatment. And you can think about the development of the cancer in the life of a patient as an arms race between the cancer and the treatment during that time. Not only would combination therapies be an excellent way, but you can think about the immune system itself as combination therapies. And we're now seeing spectacular results when we actually apply drugs that allow the immune system to do its job, recognizing abnormal peptides that are displayed by the cancer cells and killing them based on that. The thing is that when we're talking about these technologies, it's not the magic bullet that everyone has thought so much. It's not the simple pill. It's going to be a complex analysis that's required, and a lot of it personalized or precision, and that's going to require massive information exchange, just as Mike was saying. So is it hopeless that we could ever, get this smart to treat cancer? I always like to give a message of hope with discussion of this thing, and I think it's exactly the geeks in this audience, that will make the major contribution here. And why do we think it's not hopeless and infinitely complicated to figure this out? Well, one thing, if you compare cancer to HIV/AIDS, there's one thing that's really important. During this arms race, the cancer, in a sense, gets smarter and smarter to try to evade your drugs through this natural evolutionary process. But none of that smarts is passed on to our kids or their kids. So it's not really a full Darwinian evolutionary process. Each generation gets the same old cancers that its parents did. And so once we learn to play this game in a single typical individual and win it, that solution will work for our children, for our grandchildren, and so on. We don't have a constantly evolving enemy in a Darwinian sense. So we can get to this point. It's going to be very hard, though. It's not a disease that yields to any kind of standard approach. So fundamental to this is dealing with the full molecular biology of the disease, and that requires a very, very large database to get the statistical power that we can. Now, this is a cartoon, of course, and we have a number of feedback loops and algorithms and so forth in there, but at the very, very high level, the 50,000-foot level, we have just really numbers that are driving this. And one of the projects that was first to really start to attack this problem from the point of view of big numbers is the Cancer Genome Atlas, to look at 10,000 tumors in more or less complete genetic detail with matched normals, and to do this for first the 20 major adult cancers. Now it's expanded beyond that to some rarer cancers. And as Mike said, we built the Cancer Genomics Hub to deal with this first avalanche of cancer genomics data because it really was clogging the pipes. We weren't ready at the time when this project got started, and that became a big barrier. Again, beautiful points, right? We needed the data scientists at a critical time. We built CG Hub. I'll say a few things about it. One of the most important things is cost. Like, how much does it cost to really do this? CG Hub was built under contract with a specific and limited goal of storing and serving the data in the traditional way that users were expecting it. Namely, they were going to download the genomes and work on them in their home institutions. So that's the way we built it. It's 1.5 petabytes growing to 2.5 petabytes. We're very pleased that we got a high download rate. We're averaging about 5 gigabits per second, peaking at about 15 gigabits per second in terms of our download. So that we end up serving more than a petabyte of data per month to researchers all over the world. And that's actually, I checked according to the latest stats that I saw from NCBI, and we are exceeding the entire output of NCBI in some months now. So this is exciting, but just a start in terms of this. The real answer to this is more like the chart that Mike did. And honestly, we didn't look at that beforehand. But as you see these exponential growths, you get into a situation where, you get into a situation where you have data that's springing up in multiple different centers, and you cannot unify it beyond a certain point, right? So the first method, the first attempt here is, well, why don't we unify all the data in one centralized database to work with it? But you reach this critical point where that doesn't work anymore, and you have to deal with the fundamental issues of federated data. So in order to deal with that, I co-founded with 7 other researchers and scientists and individuals committed to sharing data, the Global Alliance for Genomics and Health. And this is a not-for-profit, non-governmental organization, and it's essentially like a standards-making organization. We're there to get people to work together, and we're there to try to tackle exactly the problems that Mike talked about in his introduction. It was founded in June 5th, 2013. Well, we now have more than 170 members, and we have a mission which you can read about on the web, and that is to create the interoperability we need to share genomics data, develop guidelines for harmonizing procedures for the privacy and regulatory issues, and encourage the actual stakeholders, the people that are creating these databases, to engage in voluntary sharing. Unfortunately, the situation as it is today is that as these 100,000 genomes are created, most of them are being created in medical centers that don't have a sharing plan at this point. So we lack an incredible opportunity to actually get the statistical power of large-scale genome analysis if we don't get these centers talking to each other. And this is the slide that looks a little bit like Mike's slide, and then I'll connect it with a network in a minute, right? All these same images. So we've got to do this in order to get to the point where we can really utilize these data. So how do you there's a social problem for getting them to work together on sharing, and then there's a technical problem. I'll be talking mainly about the technical problem. On the side of big data, biology is not a field that is traditionally embraced the kind of big data, large scale, large software design. And in fact, a lot of these medical centers or organizations are kind of doing the thing that bioinformatics has done a lot, hire somebody, contract it out, have your postdocs build this, and if they don't work together, which is happening all too often, then people go off and create their own incompatible pieces of software. And that will reinforce the barriers. So now, the Global Alliance, we want to try to create a system in which, a system of information exchange in which this sharing can occur. Why would you want to do this as a medical center? You want to think about, instead of building your own repository, possibly using a cloud, or if you build your own repository, then creating a cloud on top of it that's compatible with other cloud resources so people can come to you and compute next to your data. Those advantages of large-scale cloud computing are, of course, numerous in terms of, if you go with the commercial ones, we have an enormous amount of invested dollars in creating robust systems, proven security, and the basic issue of the cloud always comes back to elasticity. So it's, it essentially costs the same, about $1.50 per machine per hour, if I'm using one computer for 1,000 hours or 1,000 computers for 1 hour. And that's impossible to achieve in a single small setup economically. And then, of course, the globalization will allow us to do the globalized sharing that we want to do. So the Global Alliance is just getting started, and I want to recruit you all. I want to encourage you to get involved in this. The way it's organized is you can just go to the GitHub site here, and you can see something like 175 threads that are discussing various technical arguments about how the APIs of the Global Alliance should be constructed, and making minor refinements and tweaks and so forth. It's a completely open source environment, and there's no barrier for anyone to start into the conversation at this point. Decision making is done by the Apache Open Source Foundation rules. I don't know if you've ever worked in that kind of a global software environment. People read it, the experts read the thread, they start to make a decision. Do I want to make a pull request to pull in this new version into it? You get 3 plus 1s without a -1, and it's done. You know, -1, you got to continue talking about it. The leadership, it's been amazing. I really have been amazed to see this happening. It's so much different than working in a small university setting or a local company setting. To be working globally and openly like this is fascinating. And very naturally, among the different task teams that are developing the different APIs, the leadership just emerged out of contribution. And everybody respects those that contribute, and they naturally become leaders of that group. And we have a very simple mantra in terms of how do you get people, private companies, not-for-profits, all of these different people to work together. We collaborate on the interface, but we compete on the implementation, and that makes the companies very happy at this point that are involved. And I think it makes a lot of the data scientists that are at educational institutions and so forth also happy. So the design of it is your typical modular design, the usual diagram here, but I think it illustrates some of it. You've got these repositories that are set up with an implementation of the APIs so that they can be major servers of the data, and then you've got these application programs, clients that run on them, and they're very modular. So any one of these can run on any one of these, and we enforce interoperability. Now, what are these client apps? Let's look at beacons. This is one of the driving projects of the Global Alliance. This was Jim Ostell from NCBI's idea at the Global Alliance meeting in London about 6 months ago. Jim said, let's start with the very simplest information interchange interaction we can possibly think of and make it global. And he said, how about just asking the question, do you have any genome in your database that has an A at position whatever on chromosome 3? And the answer, yes, no, or I refuse to answer. Very, very simple. And so there is now a whole suite of methods that are trivial to get started and set up, and people are setting up beacons. And we have a so-called beacon of beacons that Mark Fumet up in Ontario has done. And so each one of these information sources has very easily set up a server for their database, and now one can ask a query like this and simultaneously query all of these beacons that are alit and get answers. One of the things that Jim emphasized when he first proposed this is he said, this is actually more a social exercise than a technical exercise. I've made it as trivial as possible from the technical point of view. The issue is, if you can't or refuse to answer that question, then why? So what policies of your institute are blocking you from answering that question? And can we rethink these? And so it actually has stimulated an enormous discussion about privacy, about sharing. Having something concrete there has been a great start. We'll get back to beacons in a minute, and the other layers that we're thinking about adding. But I want to take this as an example of what the vision of sharing would be. If you think about the internet itself, and we're really trying to build a genomic-specific language and application area, essentially, on top of the internet, there's three major things. You need a way of naming objects, you need a protocol to get them, and you need semantics that tells you what the object means. On the internet, these are very clearly defined. A URL tells you the name or identifier of an object. HTTP is a protocol for getting it, and HTML defines the semantics of what you get, what part of it's text, what part of it's an image, and so forth. The fact that we have globally agreed on all of these makes the internet work. Now, what we need to do is adapt this more specifically then to our field, to genomics. And so the Global Alliance is trying to, as a global effort, encourage everybody to work together to create these APIs. I'll tell you more about this in a minute, but we're trying to get a notion of identifiers that are content digests. This is essentially a hash function that uniquely names a data set. Then we're working on APIs that are expressed as methods. It's basically RESTful APIs for your typical operations in terms of exchange of information around the internet. For example, search reads and get a response. And then finally, what are the objects? And this is where the semantics of our field must be jointly specified by people. So what precisely is a read group? What precisely is a genetic variant? How are they specified? And each of these is specified in a JSON-like derivative. We use Avro, which is kind of a little richer than JSON, but essentially luckily there's a pretty universally adapted data definition language that's language independent. You can use whatever you want, but fundamentally everything boils down to JSON data schema underneath that. So I'm going to talk to you a little bit about globally unique identifiers, and Matt Massey is here from Berkeley, who was the inspiration of this, and Matt, you have the first implementation of this, I think, working as well. The idea of a globally unique data content digest is the following. We have, through API, semantically defined an object. So that object can actually have different manifestations. So think about it as a set of reads. It might be manifest as a BAM file, or it might be manifest as a data table that has exactly the same semantic content. Those objects will hash to a cryptographic hash string of sufficient length to be random and through a one-way hash function in such a way that this hash function will be the same for these two things because they are semantically equivalent. It will be different for any things that are semantically different. So it's unique to the data set. It's unforgeable if you give me one of these objects and put the wrong hash associated with it. I can tell you that you're lying because I can rehash like an MD5 checksum. It's privacy preserving because it's one way. You can't get the content out of it. And it's generalized in a decentralized way. We don't need a central authority to generate them. You're working in such a big space that a collision is extremely unlikely in a decentralized world. So that function of hashing generates the decentralized, privacy preserving, unforgeable, and unique properties. The content-free is because it's not computed on a specific representation of the data, but it's computed on the semantic content of the data. And then you can imagine these things being computed on coarse-grained, immutable data objects and then composable to create hashes for larger objects. So an example here is that if you think about a read set, that can be represented as an abstract object which has the semantic properties of BAM. Each of these is a read property. And you can have hashes of the different aspects of the reads, which we would call a record-level digest. So these are hashes. And then you can hash the hashes, essentially, in this blue arc to get an overall hash for the entire read set. And that gives you that composition process gives you a compound and robust hash for that. So putting all this together, we end up with a compute environment in which we can think about running an arbitrary pipeline that adheres to the API standards on any platform and create a completely portable compute environment. Now, this may be a Docker or other platform that's, again, complete industry independent, right? So you shouldn't really care about whether you're running this on Amazon or Google or your own home cloud, say OpenStack Cloud. The data environment of repositories can be managed through being a certified, essentially, implementation of the API, and we have now we're building various components of a test suite and certification process which you can go through and make sure that your implementation satisfies the requirements of the API. And that repository would also be responsible for keeping track of authorization in their own way. We're not going to dictate everybody's authorization strategy, but provide some common guidelines. Everything is exposed through the API, and so nobody cares about your internals, and that allows everybody to compete on these implementations. Security, of course, is a big deal. I won't go into the details. The emphasis here is on shared responsibility, encryption, and authentication and authorization at the data object level. So you may be authorized, it's not as in terms of large files, but in terms of specific data objects or parts of data that you have authorization for. And we're working on standards to describe who you are and how you get that authorization. But again, that has to be adapted to the Institute. Now, coming back to beacons, what I described is the first layer, and that's designed to be, from a security point of view, just like any other internet query. So all of the beacons that I just showed you have the property that anybody in the world on the internet can ask this question and get a yes or no answer. And they are functioning as anonymous users at that point. If you want more information about the provenance of that data, did it come from a specific type of medical sample that was generated at your institution at a certain time, those start to become privacy issues. And so there's a level in which you're just a registered user. You declare who you are authenticated, then you can get a little further information, and this should allow you to decide whether this data is relevant or whether you want to dig deeper. And then finally, if you really want the whole genome, that is clearly something that, for most purposes, unless you're working with the Personal Genome Project or some project that's completely consented for total unrestricted use, you will have to enter into some kind of, say, click-through contract to get access. So the working group of the Global Alliance tries to solve these problems and create these driving applications, and they separately try to meld these four areas: genome data, clinical data, security, and regulatory and ethics, and work together as a functioning unit on driving projects. I described Beacon. There's also Genomic Matchmakers, which doesn't have any open public thing, but this is the motivation here is mom walks into the hospital with a developmentally delayed 2-year-old and says, "Doc, can tell me about my kid." Kid gets sequenced, and they find a particular piece of chromosome 17 is missing, and she asks, "Doc, what about this?" And right now, the doctor says, "Well, I've never seen anything like this," because he's very unlikely to have seen anything like that in his clinic. So what do you want to do? He wants to query around the world. But clinical geneticists want this to be a secure process, and so there's a whole handshake operation in Genomic Matchmaker where you can say, "Have you ever seen any patient like this?" And when the network is fully answered, then you can be put in touch with a researcher in Tokyo and a researcher in London that also have cases, and then you go from there. It's a nice setup, and we got a number of major databases involved, and I won't have time to go into it in detail, but this is kind of a for professionals beacon project, essentially. And then the BRCA challenge is directly addressing how can we work together around the world to try to understand the mutations that are present in a specific gene that have significant clinical consequences. And as you know, there's a private company, Myriad Genetics, that has collected a lot of data about BRCA1 variants and BRCA2 variants and is not sharing that data at this point. And so the world's response is essentially, well, then let's start building our own infrastructure. And there are a number of groups that are doing this, and they've recently agreed now to start aggregating those data, and we're working with them to do that. So I will go through this quickly. The Data Working Group has a file formats task team. We're not abandoning SAM, CRAM, BAM, BCF. We need to evolve from file formats into APIs in a natural way over the next few years. Genomics Reads task team is doing the first API. The reference variants task team is talking about how we refer to variants, metadata about metadata, and benchmarking about benchmarking. I'll tell you just briefly more about these. We've created recently 4 new task teams: RNA gene expression task team, an annotation task team, so how can we formalize the way we associate properties with particular regions in a genome. Genotype to phenotype association task teams. This is essentially a semantics of the different types of associations between genotype and phenotype, what they precisely mean and how we can exchange information about those different types of associations. And then a containers and workflow task team, which Matt, who's here, leads and is working on the data digest that I talked about and other workflow issues. So I won't go through these in detail. Gives you a bit of feel for like what the file formats team is doing. It came out of 1,000 genomes, these great specs, and they are the coin of the realm at this point. And we're trying to push CRAM as a new standard instead of BAM. It's much, much smaller, and that translates into dollars immediately in terms of storage costs. There is a lot of work done by the READ API. The READ group who created the first APIs. We're now at version 0.5.1, and it's got a full implementation at Google, and others are coming along. There are servers, browsers, and so forth that are being developed at various companies. And there's a huge movement here at this point. Please look at the API, see if it satisfies your needs. If not, join the group, fix it. Metadata. Again, a number of different subgroups, and getting everybody to refer to different things in the same way and use the same ontologies is a very, very difficult thing. It's flexible where it needs to be, prescriptive where it needs to be. And finally, benchmarking. We got Justin Zook from the National Institute of Standards and Technology now heading this group, and they're working on a number of large benchmark sets, which we have available 24/7, harnesses for running those benchmarks, and also stimulation of these contests. Everybody likes a contest like the Dream Contests, and that gets people out there, gets the competitive juices flowing, and that's another way. And the important thing about it is after the contest, the data set is always available as a benchmark for anybody to use. Finally, the reference variation team. And here the goal is to look at next-generation human genetic reference that's not necessarily linear. Okay, so here's the bad news for all of you, or the good news. Your fantasy that we've been living with in genomics for a long time, that the reference genome, is suitably represented as a linear path for each chromosome, is bullshit. And in fact, that's not even what GRCH38 looks like, folks. There are hundreds of side paths, bubbles, if you will. We call them bubbles now. Like you look at MHC, there's one way to go through MHC, but there's 7 other ways in GRCH38 reference to go through MHC, and that's different DNA, folks. So a lot of our technology is based on this fiction that the reference genome is a simple linear object. It's not. It's already a graph, and we have to help everybody to catch up to that reality. And if it's going to be a graph, it should be a more comprehensive graph. So this is from Gil McVean's group. This is what, according to his analysis, MHC really looks like if you take all the data that we have now and crunch it into the various different paths. So the actual 7 representatives that we have are only a small amount of this information. And if you zoom in, you can think about this information down to the single nucleotide variant level as alternate paths through a graph. Now, how to get that going and how to represent variation from this object consistently is a complicated thing, and I'll drill into that over the next several slides. So one of the key concepts is this concept of monoallelic variants, and we'll say a little bit about that. So in particular, I point you to this schema, which is under the work in progress section. It's not part of the, didn't make the cut on the 0.5 API, but it is a work in progress section. Richard Durbin has architected. It's essentially a JSON-like schema for a graph data structure that would capture the picture that I just showed. And a number of us are working on how to map our DNA and use that graph structure. Mapping is a very tricky thing. And one of the fundamental problems we have with VCF is essentially that there are many, many different ways to express the same variant in VCF, and nobody ever really has written a definitive program that semantically verifies whether one VCF is actually equivalent to another VCF. It's important to have a way to normalize variation. In other words, we should have a standard way of expressing a variant and then allow synonyms for that. In order to do that, we have to agree on mapping. And the problem is people do mapping in all different types of ways. And yet we have this absolute conviction that if I have enough unique DNA from a typical human being, like give me 500 bases of DNA in a totally non-representative, repetitive region of the genome, is there any ambiguity about where that maps? No. I mean, you know this in your heart, right? There's just one place where that thing comes from, right? So we need to capture, we need to agree on some notion of when we're really sure there's no more doubt left, and then use that as a mapping scheme. And then we can extend it if we want to try to map other stuff, but map as much as we can that's obvious and uniquely mapped. So we're proposing and testing now what we call a left-right matching scheme, and the simplest one is exact match, where the flanking DNA has to exactly match. This is very similar if you think about how dbSNP sets up the definition. You can give them a SNP, and they have a left flank and a right flank, and that's where this SNP comes from, and it's a matching of those. Now, we also allow one mismatch, and you can do more than that, but one seems to work pretty well. So just for simplicity, we're looking at an exact scheme. So here's the reference genome, AGTC, little toy example, CTC, and now we want to match this. We want to match to this. I'm saying, does this T match to this T? Well, the idea is that in this case, this reference genome, every position in the reference genome comes with a minimally sufficient left context and a minimally sufficient right context, so that if you have that minimally sufficient context, then you map there. And those contexts are designed to be unambiguous. So they're long enough, so that's the only place you possibly could map. For any DNA string that has that left context, it's not possible to construct any other part of that DNA, any rest of that DNA sequence that could map anywhere else. So you can think about this as a minimal unique identifier on the left or the right. So this T maps here because of this. It matches, the AG matches the minimal unique identifier here shown in blue. This T maps here because it maps the CTC. And you can, in many cases, actually have sufficient context on both the left and the right to uniquely map one place. The cases that you don't map are cases where you have sufficient context to trigger a match from this T to this T because of these TTs on the left, but you also have sufficient context on your right to match to a different T. That conflict generates a no match. And the other thing is you could have not enough context on either side to match anywhere. So there's two conditions of no match. In all of these other conditions, you will match. So that's a very, very simple matching rule, and then you change a matching becomes a mapping. If every time you have a matching base, then you map that base to where it matches. It's mathematically consistent and provable that any DNA sequence, any position in any DNA sequence can match at most one place in a reference genome that is accompanied by a set of contexts that define a left-right matching scheme. So this is the important properties of the scheme or of any mapping scheme. No base in any possible DNA sequence query string can map to two different positions. Also, mathematically proven, and this becomes tricky in some schemes, it's stable in the sense that if I give you a piece of my DNA sequence and map positions in it, and then say, oh, well, wait a minute, I forgot there was actually an A and a T in front of that, and a C and a G after that, you won't change your mind about where you already mapped those bases. It's stability, it's a good property on this thing. And the nice part about this is if you have these nice stable mapping schemes, then you can naturally extend them to mapping to a graph. So the mathematical notion generalizes nicely. And so there's nothing about the linearity of the genome that I used in this. I don't need that linearity to do this. How big are these context schemes? Well, many of you have looked for unique, or maybe you built suffix trees or Burroughs-Wheeler transforms and so forth. So you know how big a piece of DNA you need before it's unique in the entire human genome, 3 billion bases. And of course, that depends on where you are. So in typical non-repetitive DNA, it's quite small. And in larger, in other regions, it's quite large. So if you look in here, around the 30s and 20s in this log scale, we have the bulk of the DNA. It doesn't take more than 30-ish bases to be unique, but then there are these regions that go out hundreds or even thousands of bases, and still they're not unique. So you have to go quite long to map to them uniquely. So these contexts, it's interesting. So it's not like a k-mer analysis at all, where k is fixed, right? Each position has enough context to identify itself within the reference genome. If we evaluate the scheme, where we use, in this case, we used a basic kind of match with allowing one difference or an exact match. These are, I'm not going to go into this. There's a number of different mapping schemes. The nice part about this is these mathematically provable, correct matching schemes, when compared to the reference genome mapping that's provided by the consortium of one MHC region to the canonical MHC region, in the primary path of the genome, get about 99% agreement, 99+% precision, and are able to map about 95% of the bases. So it's clearly a lot of these other bases could be mapped. When you look at them, you say, well, why didn't you map that? But it wasn't quite enough to be absolutely sure in this context scheme, and that's not such a big drawback of this. And then the cases where it actually disagrees look more like mistakes in the actual reference alignment, to us at least. And we tried to prove that by looking at misplaced genes. So we know a lot about the MHC. We know a lot about which genes are allelic variants of each other and which are paralogous variants of each other. There's a lot of repetition of genes and pseudogenes in the MHC. It gets quite complicated. Under a reasonable definition of what would be a mapping of the wrong gene, we just took 200 base pair strings and used our mapping versus the other mapping. Now, BWA maps more DNA, but it makes more what look like mistakes to us in this case. So it's kind of being overly zealous and trying to map. And I think one of the worst things we've done over these years is like, God, we got to try to map all these bases. But when you start screwing up that mapping, it really screws with your program. So it's better to have something that's very conservative, and then only if you want to speculate, then you go ahead and do that extra mapping. So what does this mean for naming variants? I'm finally going back to monoallelic variants. This is the reference structure now. I call it a reference structure, not a reference genome, because it's what you usually think of as a reference genome. And in this case, it's very simple. It's linear, A, G, A. But each position comes with a left and a right context, which tells me what is sufficient to map to that position. So this A is sufficient because it only requires an A on the left to map here, for example, and so forth. So these can be sets of things, too, and we'll see why that comes out in graphs. So in this case, we have a query that looks a lot like the DNA, but this position doesn't map. Well, this position doesn't map because its base itself doesn't agree. It would map, and you'll see in a minute, if there was an alternate path for this that was a T, then it would map to that alternate path. But right now, it doesn't map anywhere. And so what's going to happen is every one of these bases will either map uniquely and provably positively to one place in the genome, or it won't map. So you can parse this query sequence into bases that map, which then we have essentially, there's a universal identifier for that position in the graph. You can name it however you want, hash, whatever. And they can say, that is an instance of that variant. It's something I already knew about. It's a variant that I knew about in the human population. And then whenever you get a run of bases that doesn't map, you can think about it as a bubble. So imagine that every one of these arrows was a merging operation, and you just merged it in. This would leave a little bubble out here, like we see in the current reference genome. If I wanted to add this as a new variant to the reference genome, I would get a bubble. That bubble is a variant. Okay, that's the way I would do that variant. And if anybody else in the different part of the world came along with this sequence, Right now, they could use their own mapping and call these variants completely different, but if they use the canonical mapping, if we can agree on it, then they'll call that bubble exactly the same as a variant. So the point is, any run of unmapped bases that connect into the graph at a certain side of a certain position on one side and a certain side of another position on another, like this is position 2 on the positive side, position 4 on the negative side, where this run of bases connects, and this is the run of bases, that's a canonical name for that variant, the same name somebody in Singapore would use if they saw this query sequence and wanted to name that variant. It can be multi-base. This is a very simple example where it's just one base, and it can skip over an arbitrary part of the reference genome. So it's important that it's a universal way of defining this. And we can then collect these over a period of time. And after we learn that we're making extensive use of the same extra variant, then you can consult with the Genome Reference Graph Consortium can consult again and say, well, maybe we better add a path in the reference graph so that people don't keep naming this other variant that we left out. And the graph grows. But it grows monotonically. It doesn't shrink. So whatever mapped in the old days, as long as it had sufficient context in the new map, will still map in the new days. And that comes to the other problem, how one redoes this whole thing when you learn more about the human genome. So this is the case where we saw before. If we'd had a T already in that genome, then this thing would have mapped as an alternate path in this part here. And here's a case where we have an alternate where we have a deletion of this, and this one, of course, maps nicely because it just skips over that. And so we have in this case, we have a thread for this genome, or this haploid segment, through the entire graph without needing any bubbles or new variants to explain that. If you had a diploid genome, you would get 2 threads with bubbles in them, phased. So actually, if those of you that are VCF freaks, this is actually easier than the way VCF deals with heterozygous sites and calls. That gets really messy, and those damn little slashes and stuff, and am I phased with my next? I mean, VCF drives a number of us nuts. This is actually simpler, and the concepts from VCF are the ones that we've made into the API. So the most fundamental concept is a call. That's a single variant. Each one of these is a data set, and each one of these is a variant. So in this data set, we make a call that this variant occurs. Then a call set, of course, is all the calls for that data set, and a variant is all the calls for a variant, and then a variant set is this whole thing. And so there's a very logical hierarchy, and we don't going into the diploid site-based world from this is an algorithmic thing that gets a little more complicated, but is something that is doable when you start out with a solid base like this. So actually, monoallelic variants solve a problem because it turns out after you've used VCF for a while and the other format, actually lots of classical genetics, you realize that it's a fiction. When the genome isn't exactly diploid, then there isn't a really meaning. And when structural variants that are multi-base variants overlap, what site is variant, how, and it turns out to be inconsistent logically, and this is logically consistent. So summary approach is that, any possible phased human genetic variation relative to the reference structure is unambiguously represented. The mapping is a function, and it's stable in the sense that I described it, and the scheme is updatable. So as I alluded to, when you add more to the reference genome, you can determine what mappings have to change and what mappings don't have to change. And if that's a small fraction of the mappings that have to change, then it's a faster update than everybody rerunning BWA every time a new version of the reference genome comes out, which is, as you know, a major obstacle in terms of compute time right now. We're all suffering from that. You can get structural rearrangements that are, so you can design a browser that starts to get at this graph-like thing. So one of the things I always say, like, I'm shooting myself in the foot, right? So I've made my whole reputation on a browser that assumes this nice linear coordinate system, and now I'm saying it's a fiction. It's like, oh my God. And how are we going to change all of this stuff? So we're working on that. And so you can think about how a query sequence aligns to the genome. In this case, there's some missing, there's some data that's not there, and there's an inversion. And so you get these little, what we call snake diagrams, where these bases align to the reference like this, and then the next one goes over here, and then it goes backwards in reverse complement, aligning here, and then it continues over here. And this is actual MHC. There is a segregating inverted piece in our MHC. And so it depends on which haplotype you have in any haploid analysis of MHC, how you will align. So we can, in principle, represent these ideas. It gets a little more complicated in terms of browsing and our conceptual thing. You can, of course, in genomes have things that aren't any chromosome. So this is cancer. This is our work on glioblastoma, where you actually see in glioblastomas these small circular chromosomes that are made, chromosome-like objects that are made from different genes, and they often include key oncogenes, like EGFR is one of the accelerator petals in a critical signaling pathway that drives the cancer growth. And they have a circular, this particular GBM from TCJ has a circular chromosome-like object of about 1 megabase that has 80 copies of this cancer driver here. MDM2 is also a cancer driver because it's a gene that inhibits P53, so if you have extra MDM2, you're essentially shutting down P53, which is a favorite trick of cancer. And we have a couple of copies of it on this. So the fact that we can express such a complicated alternate version of the human genome with this notation is helpful. We will to really express what's going on in cancer, we need a general notation. Forget it if you think you're going to get by with point variants or some simple nomenclature for saying what's actually happening in these genomes. It's got to be general. Here's another double minute case from these are from a publication we made a little while ago of the analysis of the TCJ. They said, as it says here, about 20% of the GBMs have this. This is a completely different one, and this is Cameron Brennan's validation of it by FISH. This background, this pinkish background, is all probes that are lighting up to this part of the genome. So it's in many, many copies in each cell, whereas normal things are only in 2 copies or so. So altogether, just to wind up here, you've seen this a million times before, but I put it up because it's true. We want all these data out on an API, and you can design the API. Please be part of it right now. Make sure that it works for you. That API can be how you get your information, and it's made for computers and not for people. So many biologists think of point and click, and that's not where it's at. It's all about designing that API that will get you the information you need in the precise semantic form that you understand and that your programs can work on. Once that's defined and people start sharing data over that, then we can think about how we can do this virtuous feedback loop. And everybody has said a lot about this, that if you think about cohort sizes for research, we got 5,000, we got 10,000, that's a huge research project. Have you ever heard of a research project that has 1 million probands in it? No, and there never will be. But clinical practice, routine clinical practice, that has data sets on the order of millions. That's where our statistical power is coming from. So we have to get this so precise and so universal and so ubiquitous that we can actually plug it into clinical practice and let that information flow back into research in a virtuous learning cycle. That's the key. And in order to do that, we have to address this key challenge of how to normalize the exchange and sharing of genetic information. And then we have to deal with the interpretive challenge, right? Obviously, most of you work at the interpretive level. And that's fundamental, but cannot happen without the power that's provided, by this infrastructure level. And so that's why I've spent the whole lecture concentrating on the need to build that infrastructure. So I want to thank all the members of the Global Alliance Working Group, and those with a star behind them actually contributed slides to this presentation. We have, in particular, Cassie Dahl and David Glazier from Google, Benedict Payton from my group. We have a number of other co-chairs and organizers. Richard Durbin also contributed, my co-chair of the Data Working Group. There are a number of people that are involved in driving projects and are senior advisors to it, including Angel and Bob, who contributed slides to this. And I mentioned Matt, who is incubating the containers and workflow, also contributed slides. It's an exciting organization. Some of the material that I presented was presented from my lab outside of that. These are people that I collaborate with. This is my cancer group who made a lot of things. Linda Rosewood heads, for example, CG Hub. And I really am appreciative for you listening to me. Thank you. Yes?
Thank you very much for that. I think you sort of tangentially touched upon this, but say you're going to do, thank you. You want to do something like a meta-analysis, like in the GWAS style, where you can run some sort of gene burden test across these very federated data sets. So you need to ensure that every center runs a similar algorithm, and then that you can meta-analyze. Is there any work being done around this, or are we concentrating first on just making sure we can talk to each other?
Yeah, we're starting with the semantics, and then looking at how, if people are willing to put up some kind of cloud computational facility so that you can compute next to the data at their center, that has to be made easier. We're not doing that for genomics because the whole world is doing that for everything. And so OpenStack or something like this, to the extent that it gets easy to set that up. And then in terms of the actual units, so you want a self-contained unit that will run the same way in any environment. Virtual machines were, I guess, a bit heavy, and Docker is a little bit lightweight. And so, again, but this is just industry-wide stuff. We're not doing anything special for biology there. We're just using industry standards. But everybody has the same problem, that they want to reliably run their code in different places and have that, not to have to care about the details of that. And so we will adopt industry standards for making that happen. What's hard for biology is getting the information to be semantically clear and equivalent. The worst part about this thing is doing this aggregation is batch effects, because somebody collected the data one way, so it's missing metadata, batch effects because they're not really semantically equivalent in some way. And so that's the stuff that only domain experts can really sort out.
I think I'm going to use my privilege here.
Okay, yeah.
I'm going to ask a question. So I'm very interested in the Global Alliance and kind of all the great works that you guys are putting together. Now, if I was interested to participate in this, it seems like most of the members are institutions rather than individuals. Is there currently the actual membership is an institution, but membership is not an obstacle for participating in our GitHub process. So it is not required for your institution to be a member for you to participate in this. Well, I'll sign up right away.
But I would really strongly, and I did strongly encourage Bruce that Cold Spring Harbor should sign up.
Thank you.
Yes.
So one thing that I think maybe you didn't address very much, and neither did Mike, is about education.
Education.
So how do people know that APIs are a good idea? And this new proposal with the reference genome, it looks pretty complicated.
Yes, it is.
And certainly maybe less intuitive than linear sequence and slide this thing along.
Right. So
how do you get people to work on this and innovate it and not think that it's some crazy difficult thing and make it look a lot more accessible?
You first have to get it working, then you go out and train people how to use it. Our experience with the browser is that you can put as many tutorials online as you want, but you also have to reach out and actually give live tutorials. And I see James shaking his head. He does this with us for Galaxy. Again, Galaxy is an amazingly easy and wonderful workflow tool, but You have to reach out and actually say, okay, now we're sitting down, look on your monitor in front of you, we're going to build a Galaxy pipeline, or we're going to do, and then they get this hands-on experience. So it's a combination of making your best tutorial materials online and going out and face-to-face, yeah.
So actually, I want to ask you a question about one of Mike's slides.
Yeah,
which is, no, but I'm going to ask you the question.
Oh, me about my
Yes, yes. And so he had a slide with all the genome centers across, or all the sequencing machines on the map.
Right.
And if you look at it, so presumably there will be a lot of compute infrastructures at the same places. That was one of your slides.
Yeah.
But places like South America and Africa are sort of kind of thin in that space. Is the internet and cloud computing is not going to be free?
No.
So is that going to, are we going to have two worlds?
Yeah, we have to think about the inequality of who can afford to do genomics. And so the best thing to do is bring down the price first as much as we can. And I think widespread use and consumerizing all of this will bring down the price. That helps. And then you have to think about special grants and special dispensation for that. But one thing, I don't think we can assume that the sequencing machines and the computing will be actually physically near each other necessarily. There's one transfer of the data out of the sequencing machine into the cloud. It doesn't necessarily have to be your local cloud. Yes, it costs you more to physically house it far away, but that one transfer is not the fundamental. It's all the accesses that are going to come later. So I think it actually is going to be typical that the information is stored quite distant from the origin of where it was sequenced. Yes?
Hi, yeah. This is Dennis. I was wondering if, what are your thoughts on the social aspects of sharing data? As both Mike and you pointed out, the data is siloed right now.
Right.
And no doubt the Global Alliance will do a fantastic job of solving all the technological challenges around.
I'd love to
So,
you have so much faith.
So how do we convince people to actually, because I have a skeptical view of human nature, and I suspect that it's not technology that's preventing the sharing.
Yeah.
So how do we construct incentives?
Right. Well, there have been a number of proposals, and none is actually carried the day. We're feeling our way through this problem right now. The problem is people who mooch off the system, essentially. They want to download all the data, but they're not willing to contribute their own data to the pot. So so how you can create incentives or disincentives for people to do that, and incentive for them to play along is one thing. I think one of the most powerful forces on the planet are patient advocacy groups, and there are thousands of them at this point. And when they get their patients fired up, miracles happen. So the power of shame, the power of why don't you share, or the power of why can't you give me my data so I can share, you asshole, right? There's a lot of that that's brewing, and so I think those that don't play along will eventually be, there'll be a way to go around that.
So I think there's quite a lot of, certainly that's the experience we have, where everyone defaults to the highest security levels, the biggest walls around you.
Right.
Even though, for example, for us, our goal is to share the data.
Yeah, absolutely. And most of us at larger medical centers are, for good reason, caught in compliance issues that were there for good reason. And I should say it's irresponsible to go around coercing people to sharing their data in an unrestricted way if they don't understand what the consequences of that are. So the part about responsible sharing is a key thing. And with a global dialogue, I hope we can come up with some nice, fully thought out proposals in which people can be educated, know what they're getting into, and choose to share the data. I think when they know all the facts and all the benefits of it, many of them will. But no one should ever be coerced to share. Absolutely.
So I think you've totally convinced me that data federation is absolutely necessary, and that's great. At the same time, I find APIs very scary. because this reminds me of back when web services were going to be the solution to all of our data federation problems in bioinformatics. And Gunnar, the student of Gunnar's a few years ago, published a paper studying this, and there's many problems with web services, but here's at least 3 I hope you're thinking about and maybe you can tell me about. 1, the endpoints disappear. 2, the endpoints lie because you know nothing about whether the implementation behind the API is actually of decent quality, and you have no way to verify that. And 3, they can't be reproducibly queried over time. So it really hurts your ability to go back and re-ask a question. So I don't know if you're thinking
those are all
about
good. Those are all good points, and we do stress a lot about that. Those that are in the business of being reliable repositories like NCBI and EBI, who are major players in all of this, constantly stress the fact that being a reliable archive is a lot more than having a cute API. Right? So we get that every day, and for good reason. So it's not clear that just letting the world loose and letting everybody start serving data is the right thing. The hash is great in the sense that if you have an object of a file of reads and there are 52 copies of it around the world, unless all 52 disappear, then you haven't really lost it. And you can't fake it, right? You can't say, oh, I just misnamed it, and so you can't pollute the library of data because you can't fake the hash function. So there's some technical things that mitigate parts of those problems. But certainly for archival data, especially when you have versions, right? Each version is semantically different. It has to have a new hash, and then you have to have somebody keeping track of what the previous version was and what the relationship between them is. And that is an important activity that we are not claiming that the Global Alliance will take on. We'll try to build APIs so someone else can take that on at this point. And it's not clear who, who if not NCBI, EBI, I mean, the current big players.
Okay. First of all, thank you very much for the information. And actually, I have a question about the graphical reference genome. So since you mentioned it's not linear, I'm kind of surprised, actually. So I appreciate if you could share, so when and how did people to realize that the reference genome is not linear, and according to a new reference pattern, so if there's one mutation, I mean, if there's one mismatch, so as a consequence, the following sequence won't be mapped because it's kind of left, they don't have the left match. So how do you address that problem?
So if you have if you have a long sequence that matches some path in the existing reference graph, and then you have one place in it that's new that doesn't match any, there's no alternative path for that one base, then that query sequence, every position in it maps except for that one. And that one that doesn't map is a named alternative. It says, I am leaving the reference structure here, I'm seeing this novel DNA sequence, and then I'm returning to the reference structure here. So it's not like it's lost, it has a name. There's a canonical name for it that you can write in your scientific paper, you can communicate to a machine, you can communicate to a friend.
So when you get the sequence from sequencing machine, it's kind of linear.
Yes.
In this new model, then you won't do it from the first base to the second base, third base, like consecutively, but maybe you need to get some random pieces from the sequence.
There's an issue that I swept under the rug with noisy genome data directly raw out of a genome sequence. You really need to do some kind of assembly or noise reduction on that before it's going to map decently. So there's an iterative process. So the current mapping schemes we're exploring are actually mapping contigs, essentially. And you may get those contigs either by de novo assembly or initially by some heuristic mapping, not the canonical mapping, but you can, but BWA, I don't say anything negative about BWA. BWA is a great thing, and there's even a cooler one, Snap. It's 10 times faster, and Hang Lee was saying this is pretty cool. So that's great, but once you get to the point where you've assembled with high accuracy a fairly large sizable piece of DNA, hundreds of bases, like we were doing them with 200 base hunks, then that's enough to already get this fundamentally unique mapping that no one would ever argue about to most places in the genome.
Thank you very much.
Yeah, so the example of is there a base which is A at this position is a very good one to choose because it's one that current implementations like VCF tools would take a very long time to implement. In thinking at some of the other things in your API, I was curious if there are plans or could there be plans to have benchmarks which address similar questions. For example, if I want a 1 million base pair chunk of mappings that match the reference genome in CRAM, in BAM, delivered by SAM tools, and in your API, are there plans to have benchmarks for how long that's going to take and how much data is transferred?
So an extremely important part of a benchmark is how long it takes to run it. And that we intend that to be a fundamental part. So it should give the right answer in a reasonable amount of time. And that has to be done on in a standardized environment. For example, not to say that Amazon is the solution to everything, but many of you use Amazon, right? If everybody runs the benchmark, or Amazon agrees, and they have said that they would do this, to allow people to run competitive benchmarking on the same configuration, then the time means something because the compute environment is identical and we can compare how long it took to do it under a certain resource. So that should be a part of the benchmark, absolutely. And I would say one more thing, the APIs that we're designing are starting at the simplest level about communicating reasonably small amounts of information rapidly around the world over the net. It's a different problem. The intercommunication links that you need inside a large fixed computer when you're doing a compute-intensive calculation, you may not want to use the API for the so-called over-the-wire transmissions. And so there's a Hadoop extension that people are working on. So this would be basically libraries that you would build that would implement equivalents to these things, but in an environment that is a power environment inside a big center. So there's another criticism. I thought that's where you're going, that it's too inefficient to always use this API. What if I'm crunching 10,000 genomes inside my center? Well, then you'll have a souped-up, hyper-efficient version of this that's certified.
Maybe one last question up front.
So I just wanted to follow up the response on the API comment, where all of the disadvantaged APIs or problems that you mentioned are all related to federation and networking. But I imagine that a lot of the actual computation is going to happen within your own data center or on your own local machine. Yes. And APIs are still highly valuable in that case as well. For example, if you're, BWA should not be reading a file format. It should be reading from an API and completely abstract away all of the underlying storage. And so this would essentially solve the problem that bioinformaticians spend an enormous amount of time dealing with file formats and converting them. Right. But APIs are already useful even without thinking about the network issues.
Thank you. The convert. No, that's absolutely true. Thank you. Yes.
Okay. David, thank you so much for an excellent keynote this evening.
Thank you.